Hello, everyone. Welcome to SRNT's 2021 awards program. I'm having some internet stability issues, which is why you can't see me on the screen. Before we begin, I'd like to sincerely thank all of you who nominated your colleagues and created the slate of nominees for all their incredible work in nicotine and tobacco research. I'd also like to thank this year's award committee, which is chaired by Tom Glynn and includes SRNT's three presidents, myself, Suzanne Colby, who's the past president, and Megan Piper, the president-elect. And we were also very fortunate to have Laura Beirut join our group this year as a past winner of the Langley Award herself. So thank you to everyone for all your work. And now it's my great pleasure to introduce Tom Glynn, the chair of the 2021 Awards Committee, who will introduce the awardees. Tom, take it away.
Thank you, Suchitra. And I'll add my good morning, afternoon, or evening to all of you, and welcome to the 2021 first and hopefully only virtual SRNT award ceremony. I too want to thank all those who nominated someone for an award and the award committee itself, Suchitra, Suzanne Colby, Megan Piper, and Laura Beirut. And thanks as always to Bruce Wheeler, Nan Knudsen, and Brooke Miller at Rees Group for making this process run so smoothly. Being on the awards committee offers both pleasures and challenges. We have the pleasure of seeing the astounding accomplishments of many of our members, and at the same time have the challenge of selecting the final awardees from among the many, many superbly qualified nominees each year. Our hope is that as in past years, we have represented SRNT well in presenting to you this year's slate of awardees. I was getting ahead of myself there with Slade. We have awards today in four categories: New Investigator Best Abstract Awards, the Jarvik Russell Early Career Award, the John Slade Award, and the Langley Award. And detailed descriptions of these awards may be found in your online SRNT program. So let's begin. The New Investigator Best Abstract Awards are just that, annual meeting abstracts from new investigators chosen by the program committee to be the best among the many outstanding abstracts received from newer investigators each year. This year's awardees are Jamie Hartman Boyce for Update on the Cochrane Systematic Review and Meta-Analysis of Electronic Cigarettes for Smoking Cessation, Carrie M. Rosario for The Elephant in the Room, a scoping review of naming racism in tobacco control, Yasmin Sharifat for the Nicotinic Receptor Modulator Links 1 in nicotine reinforcement, and Andrea R. Titus for Smoke-Free Laws and Disparities in Secondhand Smoke Exposure among Non-Smoking Adults in the U.S., 1999-2014. Congratulations to each of you, and we look forward to more great things from you in the coming years and to your presentations. Our next award is the Jarvik Russell Early Career Award, named for tobacco control research pioneers Murray Jarvik and Michael Russell, and which recognizes scientists who early in their careers have made extraordinary contributions to the field of nicotine and tobacco research. This year's Jarvik Russell awardee is Dr. Gillian Gould of the School of Medicine and Public Health at the University of Newcastle and Hunter Medical Research Institute in Australia. For those of you fortunate enough to have seen Gillian deliver her Jarvik Russell New Investigator plenary yesterday, and if you didn't, I highly recommend going back and accessing it. She aptly described her career as a journey from being a dedicated GP delivering clinical services to a researcher investigating the most appropriate ways of working with Indigenous peoples to aid their communities in stopping smoking. Gillian has had a wide range of research support in a relatively brief time, has published widely, and received numerous other awards. Her nomination letters for the Jarvik Russell Award were truly outstanding and noted her distinct and unique ability for nurturing reciprocal relationships with the communities with which she works. I'll close my introduction to Gillian with two comments. She is, I think, the only Jarvik Russell awardee to have among her many degrees a diploma in drama. And she has also traveled, albeit virtually, the furthest among our awardees today, 9,788 virtual miles from Callahan, Australia to Baltimore, Maryland, USA. Gillian, congratulations again, and please just say a few words for us.
Thank you. I would like to express my profound appreciation for the honor conferred by this esteemed award named after two pioneers of tobacco research, Murray Jarvik and Michael Russell. This is the first time I have received such international recognition. I acknowledge that I have not gone down the standard career path for a researcher, and that can lead to a bit of ambiguity. My previous clinical and public health work among Indigenous populations for tobacco definitely gave me a head start to my research once I commenced my PhD in 2012, and even more so since finishing in 2015. My major thanks go to the Indigenous health services, health professionals, and Indigenous experts who have supported my research endeavours and those of my team and PhD students. I would not have been successful on my research journey if it were not for the very generous and forgiving nature of the majority of Aboriginal and Torres Strait Islander people I have met, because working across cultures means inevitably mistakes are made. They have inspired me, and in turn, I hope I have inspired them with my vision, and we have walked together in allyship. I thank my nominators, Professor Amanda Baker and the late Associate Professor Marie Gruppetta. The latter was my unerring supporter and Indigenous mentor, and I miss her more than words can say. Marie would have been proud and happy today to see how this work is going forward. I thank all of my team, past and present, and those who have sustained their interest and helped me build capacity in this rewarding and challenging field of work. My collaborators are numerous, and I appreciate how each have brought me something valuable at the right time, so I am able to go on being creative and scientific in equal measure. I am immensely pleased that I have so far had continuous funding from the National Health and Medical Research Council Australia since 2012, and from Cancer Institute New South Wales since 2016. But thanks to all of my funders for funding me to a small or large degree, including my GP professional college, the RACGP. And lastly, I thank my family and friends for being overwhelmingly supportive. Without them, none of this could have been possible. Thanks.
We continue now with the John Slade Award, which honors individuals who have made outstanding contributions to public health and tobacco control through science-based public policy research and public advocacy. This award is, of course, named after our late SRNT member and researcher advocate extraordinaire, Dr. John Slade. Due to the COVID situation, and I'll leave it at that overlaid last year's SRNT meetings in New Orleans, the 2020 Slade awardee, Ron Borland, was unable to come to New Orleans to receive his award. But as a demonstration of SRNT's ability to cope with adversity, Ron will receive his award this year, and following Ron's acceptance of this award, should he choose to accept it a year late, we will then go on to the 2021 award. So back to 2020, not a pleasant year, but here we go. Dr. Ron Borland is the Nigel Gray Distinguished Fellow in Cancer Prevention at the Cancer Council of Victoria. He has deep roots in psychology and has used his training in that field to conduct meticulous tobacco control research that he then helps policymakers understand, use, and implement. All qualities that, as one of Ron's nominating letters noted, would have made John Slade enthusiastically concur with bestowing this award on Ron. Ron's work in tobacco control research goes back several decades, and his influence has been felt far beyond his native Australia. Throughout his career, Ron has sustained a national and international reputation as one of the most influential tobacco control scientists in the world. As the author of more than 400 scientific papers, books, and chapters, and a key presenter at numerous international scientific meetings, Ron's work and the work of many scientists he has mentored has helped shape population-based tobacco control policies that have been adopted by countries around the world. His most recent work as a principal in the International Tobacco Control Policy Evaluation Project, the ITC as it's known, applies rigorous scientific standards to evaluate the WHO's Framework Convention on Tobacco Control Policies as they are implemented by the 180 parties to this treaty. Ron's work and those of his colleagues on the ITC have and continue to have an enormous influence on global tobacco control. Finally, we should also note that in addition to recognizing Ron's scientific contributions to tobacco control, he should also be recognized for virtually traveling the second furthest to receive this award today, 9,273 miles. Ron, please say a few words for us.
Thanks, Tom, for those kind remarks. I'm deeply honoured and still a little surprised to have won this distinguished award. I have strong memories of John Slade, a dapper man with a snappy bow tie, the perfect disguise for a radical tobacco control advocate, and someone whose achievements I could never hope to match. My efforts in support of public policy have rarely been on the front line. I've seen myself more as an armourer for advocates and policymakers, providing good science and rigorous evaluation to support appropriate actions and to ensure we're on the right track. For the bulk of my career, I worked as a researcher at the Cancer Council Victoria, a small organisation with a large international impact. I've been supported by some extraordinary mentors, particularly David Hill and Nigel Gray. I've also had extraordinary collaborators, largely those on the ITC team, Mike Cummings, Jeff Fong, Anne McNeil, and many of the cast of hundreds that I've worked with directly. I've also worked with some fantastic Cancer Council researchers, including the late James Barmford, Wai Yong, Lin Lee, Bill King, Dave Young, Mohammed Siapush, Kathy Segan, and Michelle Scolo, only to mention the longest standing relationships. I see this award as a collective tribute to all these people rather than specifically to me. After all, public health research is a team sport. Being part of a team has allowed me to take a broad systems approach to my research and to not only work out what might work, but how it works, and as far as possible, to demonstrate that it works as theorised. For me, this has been an exciting intellectual journey, and it has been deeply gratifying that it has helped shape tobacco control policies and programs in a number of ways, however small. I hope I can continue to contribute to the elimination of cigarette smoking within my lifetime. Our collective work has reinforced the reality that rationality is a fragile resource, and that the challenges we all face is to ensure our passion is grounded in the strongest possible understanding of the underlying reality, not by selectively picking evidence to support what we would like to believe to be the case. This applies equally to our research as it does to smokers attempting to free themselves of a dependence many feel emotionally tied to. Finally, I would like to thank my family, Virginia Lewis, the love of my life, and my two wonderful sons. They've kept me grounded and helped keep me aware that what we are working for is for the well-being of people in general. It has been a privilege to work in this life-affirming field. Thank you again for this wonderful award.
Moving on to 2021, I am so pleased to introduce Patricia Nez Henderson as the 2021 John Slade Award recipient. Dr. Nez Henderson is Vice President of the Black Hills Center for American Indian Health, an institution that has among its goals the creation of culturally relevant smoking prevention and cessation interventions. Patricia is unquestionably the leading national expert on improving American Indian health through tobacco control. She has worked tirelessly on this issue for 2 decades, conducting groundbreaking research, advocating for policy change, and representing the American Indian community at the highest levels of national and international research and policy discourse. Patricia represents the true Sladian, if I can use that term, ideal. She conducts meticulous research and then sees that research through to translation, application, and where appropriate, policy initiation or change. As the first Navajo woman to receive an MD degree from the Yale University School of Medicine, Patricia is accustomed to being a pioneer. She has also founded the SRNT Indigenous Circle, an international network that fosters the growth of Indigenous scientists, and was the first investigator to describe the high cotinine levels in American Indian children and adults non-smokers. Patricia Nez Henderson is an exceptionally worthy recipient of the John Slade Award. And Patricia, could you give us a few words, please?
Yet a hello. I would like to first acknowledge the traditional landowners of the land in which I work, live, and play. These are the homelands of the Lakota people, Haisapa. Having grown up on the Navajo Nation, I witnessed many of the Navajo traditional healers using ceremonial tobacco as a form of healing, restoration, and overall life balance. My view of the word tobacco came from this positive context. When I began my research career in public health, that's when I began to see the differences. I remember going to a meeting in Navajo Nation with over 100 traditional healers, and when I asked who used commercial tobacco products in their ceremonies, nearly 70% of them raised their hands. It was in that moment that I realized that there was an issue, one rooted in colonization and culture erasure. I addressed the healers and said to them in Navajo, which translates to, commercial tobacco products will hurt the inner essence of who you are and everything around you. I then said to them that the plant had good intention when it was created, but man adulterated it. I was very surprised that they understood what I said in Navajo and listened to what I had to say. They affirmed my intense and loving remarks and expressed interest in working with me. Because of this experience, the focus of my career not only has been to change the narrative about the word tobacco for Indigenous and non-Indigenous communities, but to work even more closely with Indigenous traditional healers and elders, assuring that my work is informed by their perspectives. I am humbled and honored to be the recipient of the 2021 SRNT John Slade Award. I'm truly grateful for the people and experiences that have shaped me as a scientist. There are several people I would like to thank today. First, I'd like to thank SRNT for appreciating all the work we have done through science to enhance the health of Indigenous communities. I also like to thank the people who have mentored and supported me from childhood to now, my parents to my professional mentors. I'll also like to thank the Navajo elders who understood the importance of receiving an education, a Western education, all while maintaining my Navajo culture and traditions. Next, I would like to thank communities that I've collaborated with throughout my whole career, especially the Navajo tribe and the Lakota communities. Our relationship was built on trust and safety. This allowed us to create cultural relevant interventions for their communities and to spearhead policy changes with the study findings. Finally, I would like to thank my family and my team members at the Black Hills Center for American Indian Health. I especially want to thank my husband and our two children. I have spent countless hours crisscrossing the Navajo Nation and other tribal lands or on the phone with tribal leaders. And my family and my team members have always been there to support my efforts. They have been there every step of the way. Thank you. Ikeha.
Thank you, Patricia. Now, our final award today is the Langley Award for basic research on nicotine and tobacco, and is named for John Langley, who first described the concept of receptors on cells that bind transmitters and thus initiate biologic processes. The 2021 Langley awardee is former SRNT President, Dr. Marina Peixoto. Marina will deliver the Langley preclinical lecture after receiving her award, and she has asked her fellow Yale Bulldog and current SRNT President, Dr. Suchitra Krishnan Saran, to introduce her. Suchitra.
Thank you, Tom. It is my great pleasure to introduce the winner of the Langley Award. For those of you who don't know, my background is in pharmacology and toxicology, and I learned all about John Langley's pioneering research describing the effect of nicotine at neuromuscular and autonomic ganglionic sites, which really led to the conceptualization that neuronal transmission may be chemically mediated and that there might be neuronal receptors. Thus, I think it's only fitting that this year the Langley Award should go to an individual who has done groundbreaking work on advancing our knowledge of nicotine neurobiology. My friend and colleague, Marina Picciotto, who's also a past president of SRNT. Marina and I go way back. We have known each other for almost 25 years. But I think we both got really close when we were pregnant at the same time, which coincidentally was also when we had our first NIH tobacco center at Yale. Her daughter was born 2 weeks before my son, and they both went to the same daycare. So Marina and I were also nursing buddies, and we went through all the stresses of being a first-time parent together. I have learned over the years that she's not only an outstanding scientist, but a wonderful and caring person. We have shared many successes and cried over many losses together. I call her all the time with questions about neurotransmitters and second messenger systems. And if she does not answer, I even know where she lives. Sorry, Marina. But jokes apart, Dr. Marina Picciotto has been on the faculty at Yale University since 1995, and she's currently the Charles Murphy Professor of Psychiatry, the Deputy Chair for Basic Science, and Professor in the Department of Neuroscience, Pharmacology, and in the Child Study Center in the School of Medicine. Training under mentors like Paul Greengard and Jean-Pierre Changeau, she used molecular genetics to develop a line of mice lacking one of the primary subunits of the brain receptor for nicotine, which she and many other colleagues have used to elaborate on pathways leading from single molecules to nicotine addiction. Her research focuses on understanding nicotine addiction and behaviors relevant to human smoking and vaping in preclinical animal models, and then translating these findings into collaborative clinical research projects. I'm very fortunate to have worked with her on many collaborative initiatives. Marina's research is highly recognized all over the world. She has received the Human Frontiers Science Foundation 10th Anniversary Prize in recognition of her commitment to international collaborations. She was one of 60 young US scientists awarded the Presidential Early Career Award for Scientists and Engineers by President Clinton in 2000. And she was also elected to the National Academy of Science in 2012, and more recently was awarded the Carnegie Prize in Mind and Brain Sciences. I cannot think of a more outstanding person to receive this award. Congratulations, Marina. Please take it away.
Thank you very much for this award. It is an incredible honor to be the Langley awardee this year. It's especially poignant for me because my postdoctoral mentor, Jean-Pierre Changeau, was one of the Langley Award winners. And so many of the people whose work has inspired me and who showed me how to do good science and to understand the basic neurobiology of the nicotinic acetylcholine receptors in the system are past winners. It's also a real honor to be nominated for this award, and those who nominated me had to put in a lot of work, and I very much appreciate their effort. I also want to thank the many, many trainees and both the graduate students, the postdocs, the research scientists who've worked with me over the years. Science is not in any way a unitary, solitary field. It's a team effort, and it's something that has really enriched my thinking about nicotinic acetylcholine receptors, about nicotine addiction. And in particular, I want to thank my colleagues in the Department of Psychiatry at Yale University, who have taught me a lot about how basic molecular and neurobiological science can inform human health interventions, understanding of the real scourge of nicotine addiction in patients and in human subjects. Their patience in helping me to understand the language of clinical research and of clinical medicine has made it that much easier for us to take our neurobiology work in directions that are relevant to smokers and to people who suffer from smoking-related illness. And I want to thank all of you for your attention, for taking time out of your day to listen to a talk about nicotinic acetylcholine receptors. and hopefully to share in the excitement and interest that I find in these amazing molecules and that have inspired the work that I've done over the last 25 years. Thank you again. Hello, everyone. My name is Marina Picciotto, and I'm very excited to talk to you today about the work that my laboratory, our collaborators have been doing over the past 25 or so years on the role of individual nicotinic acetylcholine receptors and behaviors that are relevant to nicotine addiction and to the behaviors that drive smoking in human smokers. Before I start, I want to disclose that this work has been funded by the National Institutes of Health, the Brain and Behavior Research Foundation, also known as NARSAD, the Kavli Foundation, as well as funds from the Connecticut State Department of Mental Health and Addiction Services. I don't have other financial or other conflicts of interest. So here's the problem that we've been working on for many years. First, there is a large family of nicotinic acetylcholine receptor subunits. These are outlined here in 3 different colors that are the molecular targets for the nicotine in smoked and vaped or ingested tobacco products. And these nicotinic acetylcholine receptors are expressed throughout the brain and body in almost every cell that are in almost every cell in the brain, as well as many cells that are not neurons. And finally, we know that there are many, many reasons that people smoke. Certainly, nicotine is an addictive component of tobacco products. It drives ongoing smoking. It can be rewarding. It is certainly reinforcing. It increases the behaviors that lead to its ingestion. But smokers also report that they smoke to control appetite, or that they smoke when they're anxious or when they're depressed. And we've spent the last couple of decades trying to connect up these molecules, these areas of the brain, and these behaviors so that we can understand the molecular basis of the different aspects of tobacco smoking and vaping that drive these ongoing behaviors. So first, let me start by introducing this family of molecules. The nicotinic acetylcholine receptors are ion channels. They sit in the membrane of cells, and they form a pore across the membrane of those cells. And when nicotine or the endogenous, the normal neurotransmitter acetylcholine binds to these molecules, this pore opens, ions flow into the cells on which these receptors reside, and that activates or increases the firing of the nerve cells on which these receptors reside. And there are many broad families of these receptors. Each of these receptors is made up of 5 subunits. So each of these is a different independent protein. And when these 5 subunits that are all very similar in structure get together into a molecule, they form this barrel-like structure with a hole in the middle. That's the pore. So here's the side view, and here now we're looking at it face on. And this here on the left is the canonical nicotinic acetylcholine receptor that has been studied from the time of Langley onward. And this is the receptor that sits in the muscle. And it's right across the synapse from the motor neuron that brings impulses into the muscle, causes it to contract, and allows you to control whether you move, whether you reach for something. And these nicotinic acetylcholine receptors are clustered across from the site of release of acetylcholine. And when a molecule of acetylcholine binds at the interface between these subunits, these receptors open and the muscle twitches. We know a lot about the nicotinic acetylcholine receptors in the brain as a result of homology with this muscle receptor. And when nicotine from tobacco gets into the brain, these 3 families of nicotinic acetylcholine receptors, and they're quite broad families, I'm giving you 3 examples here, react very much like when acetylcholine is released at the neuromuscular junction. So the families of nicotinic receptors that are expressed in the brain are also pentamers. They're also ion channels. They have a pore in the middle. But one of these families, made up generally of the alpha-7 subunit, has 5 subunits that are all the same around this pore, and that's called a homomer. And that changes its biophysical properties. So instead of having, by homology, just 2 sites for binding of nicotine or acetylcholine, these receptors have 5, and that changes their properties. Then these 2 families, and I'm giving examples here of beta-4 and alpha-3, or beta-2 and alpha-4, are found throughout the brain and also in the autonomic ganglia. So the autonomic nervous system, the fight or flight system, is also regulated by nicotinic acetylcholine receptors. And in particular, in the autonomic nervous system, this type, the beta-4 alpha-3 type, allows acetylcholine to excite your nervous system or to conduct information from the brain to the body and from the body back to the brain. In the brain, we have the most widespread receptor is this that contains the beta-2 subunit and the alpha-4 subunit, as well as other subunits, and it varies depending on the cell. And these receptors are unique in that they are very, very high affinity for nicotine. What does that mean? It means that very low levels of nicotine, the low levels that come into the brain from vaping and smoking, are enough to activate and to also both activate and then turn off, desensitize this family of receptors. And so it's certainly a combination of all 3 of these families that are important for the action of nicotine on the brain from smoked tobacco products. But certainly we know that this subclass is the most sensitive to the nicotine in tobacco. So now let's get into the behaviors. Why do people smoke despite negative consequences on health? Well, we know that nicotine is rewarding, reinforcing, and it drives ongoing tobacco smoking and nicotine vaping. If you remove nicotine from a tobacco product, people will slowly over time extinguish their use of that tobacco product and will usually switch to another brand. So we know that it is essential for ongoing smoking. And like other addictive substances, we know that nicotine and nicotinic receptors excite the part of the brain called the mesolimbic dopamine system, in which the neurotransmitter dopamine has its cell bodies here in the ventral tegmental area, or VTA, and then the ends, the terminals of these neurons in the nucleus accumbens, which is part of the striatum, so forward in the brain. When these neurons are excited by nicotinic acetylcholine receptors on their cell bodies or on their terminals, dopamine is released, and we know that it is essential for the rewarding properties of nicotine for this dopamine system to be active. If you block this system, or if this system is lesioned, damaged, then you don't see the rewarding effects of nicotine. And this is a system that we know a lot about because it is a place in the brain where all of the addictive substances that are abused by humans converge. So first, we know that cocaine blocks the reuptake of dopamine, prolonging its signaling in the synapse. We know that cannabinoids, alcohol, opioids all impinge on this system in order to be rewarding and to be addictive. So the first area of the brain in which we started to look for the effects of nicotinic acetylcholine receptors when we started these studies was in the mesolimbic dopamine system. And so this is a slice through the, or these are slices through the mouse brain in which we have looked for the expression of those nicotinic acetylcholine receptor subunits that I told you about earlier in the talk. And what you can see here is that this mustache-shaped structure is the ventral tegmental area, or VTA, where those dopamine cell bodies reside. And what you can see is that there are some subunits of the nicotinic receptors that are expressed almost exclusively in these brain areas. So alpha-6 and beta-3 are really highly enriched in these dopamine neurons. And then there are these subunits, the beta-2 and alpha-4, that I told you are widespread in the brain. They're expressed throughout the brain, but we still see a concentration of their expression here in this mustache-shaped area, the VTA. We know then that there are a lot of different subtypes of the nicotinic acetylcholine receptors that are expressed in this very important dopamine region of the brain. And so when we began this work long, long ago, it was the dawn of the era when we could manipulate individual genes and individual gene products in the brain and body of transgenic mice. What we were able to do is to take that large family of nicotinic acetylcholine receptors and one by one target the genes for each of those subunits. And as a field, what we've done is to individually knock out, remove, suppress the activity of each of those different subunits. And then in even more sophisticated experiments, either remove them selectively from different cell types or put them back into different cell types and ask which of these molecules and which of these cell types is essential for the rewarding aspects of nicotine. And I'm going to show you some historical data and to show you first that this is the binding of radio-labeled nicotine in the brain of a normal mouse. You can see that it follows the pattern of the expression of those widespread subunits, the beta-2 and alpha-4 that I told you are very tightly binding to nicotine. And when we knock out or get rid of one subunit, the beta-2 subunit, all of that high affinity, strong binding to nicotine is gone in the mouse brain. So we know that this particular subclass depends on having at least the beta-2 subunit expressed in the brain. And so what happens when you get rid of the beta-2 subunit, get rid of these high affinity nicotinic receptors? Well, first of all, when we make a slice through those dopamine cell bodies and we put an electrode, basically a pipette, onto the cell bodies of the dopamine neurons, we know that normally nicotine causes these neurons to fire. So each of these ticks is an action potential that is one of those dopamine neurons firing. And when you put nicotine on, you can see more of these ticks. So more of the firing of these dopamine neurons. And if you do the same experiment on a dopamine cell from these beta-2 knockout mice, now nicotine does nothing. Basically, these neurons just keep firing as if there was no nicotine at all. So that was our first indication that you need this particular subtype of nicotine receptor in order for nicotine to increase the activity of the cell bodies of these rewarding neurons. And then we looked in the whole brain because we knew that nicotine receptors were expressed throughout the brain and body. Maybe nicotine could work somewhere else in order to get dopamine released. And so what we did was to put a pipette into the terminals, the place where the dopamine is actually released in the nucleus accumbens. And we measured all of the dopamine release when we exposed the animals to nicotine throughout the brain and body. And what you can see is that we can measure increasing amounts of dopamine released in this system when when the whole animal is exposed to nicotine. But if this animal lacks these high affinity nicotinic acetylcholine receptors, now there's absolutely no release of dopamine at all. So you need these receptors for this to happen. And finally, we then wanted to know in those terminals themselves where the dopamine is released. If we put those in a test tube and we put nicotine on those terminals, we know that we can actually get release of dopamine just from those isolated parts of a cell alone. And that's in these hatched bars. In normal mice, when nicotine is put onto these dopamine terminals, you get increased dopamine release just from that part of the cell alone. And when we get rid of the beta-2 subunit, so those high affinity nicotinic receptors, and that's down here, all of that is gone. So at each of the levels that we look, you need this particular subtype of the receptor in order to get dopamine release. And how does that translate to behavior? Well, first of all, I do want to show you that the nicotine receptors are on many, many parts of the cell. So we know that there are at least 2 or 3 subtypes of nicotine receptors on the cell bodies of those dopamine cells. They're, as I mentioned, on the terminals of those cells, but they're also on cells that express other neurotransmitters, excitatory neurotransmitters, inhibitory neurotransmitters. So it's quite a complicated system. So we could imagine that even though we've gotten rid of the nicotine-induced dopamine release through these beta-2 containing receptors, there may be other types of nicotinic receptors that could induce behaviors related to reward. And so what we looked then at is at many different hallmarks of how nicotine can increase the activity of the dopamine system and at reward. So first is movement. We know, for example, that other drugs of abuse that excite or that activate the dopamine system make you very hyper aroused and very alert. And in a mouse, this translates to moving around, to locomotion. And what you can see is that nicotine does increase the locomotor behavior of mice normally. But when we get rid of these high affinity nicotinic receptors, the beta-2 subunit, that's gone. So nicotine now doesn't make you alert. It doesn't increase your movement. What else can we look at? We can look at whether animals like nicotine. And so in this experiment, we give the animal a choice between two chambers that they can tell the difference between. And in one chamber, they just get a saline injection, so just an injection. And in the other, they get a nicotine injection. And what you can see is that in normal mice, if you do this at the right concentration, there's no preference between the two chambers when you give saline in each chamber. But if you give nicotine in one compartment and saline in the other, and you ask them to choose where are you going to explore, they will explore in the chamber where they got the nicotine. And finally, if we look at these knockout mice that don't have these high affinity nicotinic receptors, that's completely gone. So the activity and the reward depends on this particular subunit in this system. And finally, the hallmark of addiction experiments in model organisms is, will they work to get an infusion of that drug? And so you can do this with cocaine, you can do this with opiates, and animals will poke their nose or press a lever or do some action, run down a ramp in order to get an injection of the drug. And that's true for nicotine as well. And so in this experiment, we trained all of the animals to poke their nose for an infusion. In this case, we started by training them on cocaine, and then we switched them to nicotine. And if normal animals then are switched to nicotine, they continue to poke their nose. They continue to work for that nicotine. If normal animals are switched to saline, so they're getting nothing, they stop poking their nose. They don't want to work anymore. And then they just start getting down to random chance. But if these knockout animals that don't have the beta-2 subunit are switched from cocaine to nicotine, it's as if they got a saline infusion. They start giving up on poking their nose, and they get down to random chance, as if they really don't feel that reinforcing effect of nicotine. So taken all together, this first quite elderly work by now gives us a clue into what is it that is the molecular basis of the first effects of nicotine, the ones that we know are essential for people starting to vape, starting to smoke, and for maintaining that behavior. Well, we know that it is nicotinic acetylcholine receptors containing the beta-2 subunit. And from work in the larger field, we know that they also contain the alpha-4 and the alpha-6 subunit. They're expressed on dopamine neurons in the ventral tegmental area. And this subtype, tegmental area, is essential for the activating and reinforcing aspects of nicotine. So we know that this is important for human smoking because knowledge of this particular receptor subtype was essential to developing one of the current medications to help people stop smoking, and that is the molecule varenicline, which binds to this particular type of nicotinic acetylcholine receptor, excites it, but not quite as much as nicotine, and therefore allows people to wean themselves off nicotine without getting the full withdrawal symptoms that you get from stopping cold turkey, and allows people to stop smoking more easily. So I think this is one example of how the very basic research on which molecules, which cells are important for nicotine reinforcement, nicotine addiction, can be translated to a medication that can really make a difference for human health. So why else do people smoke if it's not only the reward? And certainly if it were only the reward, smoking would be a lot like other drugs, maybe cocaine, maybe heroin, but it isn't. People are not euphoric when they smoke for the most part, and instead that they report many, many different reasons that they smoke. So one thing that keeps people from stopping smoking is that smokers are leaner on average across the whole population than non-smokers, and they gain about, on average, again, people can gain much more, 2.5 kilograms, a little bit more than 5 pounds when they quit smoking. And what's really, I'd say, the most discouraging is that in some surveys, when teenage girls are asked, why did you start smoking or why did you start using nicotine products, they say they want to control their appetite, they want to control their weight. And so mice too experience this effect of nicotine. So mice over the course of their late adolescence gain weight quite rapidly. And these are mice that are, I would say, in the range of 30 days, 30 to 40 days in this experiment. And if you start in giving them increasing doses of nicotine, you can see that you blunt that weight gain, and that this blunting lasts as long as you continue to give them nicotine. So it's clear that even in mice, nicotine controls weight gain. And we know a lot about the systems in the brain that regulate appetite, that regulate homeostasis, that is the interaction between the needs for calories from the body and the drive to look for food by the brain. And the part of the brain that is most well studied around appetite is this part of the hypothalamus, which is deep in the brain, called the arcuate nucleus. And in the arcuate nucleus, there are a few different cell types, but the two main cell types are those that express different peptides that are markers of their phenotype, of their identity. So one is called NPY-AGRP cells, and if you just isolate these cells and stimulate them in an awake mouse, that mouse will go and voraciously eat, will find food and eat, even if it is not hungry usually. And then there's another population of cells called the POMC cells, and in those cells are a little more complicated, but if you stimulate them in the right context, you can decrease appetite, you can decrease seeking for food. And so when we started these experiments, we thought, well, maybe there are nicotinic acetylcholine receptors, some kind of nicotine receptors on these POMC cells, and maybe that contributes to the ability of nicotine to decrease food intake, decrease appetite, maybe decrease body weight. And so what we did was to isolate, again, those cells in a slice through the mouse hypothalamus. And sure enough, if you put nicotine onto those cells, they fire. Again, each of these ticks is one of these, is the activity of one of these POMC cells, the appetite suppressing cells, and nicotine can increase their firing. And then if you look at the doses, as you increase dose, as you might expect, you get more firing of these cells. So then what we did was, rather than getting rid of all subtypes of nicotine, all subunits of the nicotinic acetylcholine receptor throughout the brain and body, we used a molecular trick, which is using what's called a small hairpin RNA. And these small hairpin RNAs find their match in the cell, they bind to their match, and they decrease the translation, the making of the protein for which they are targeted. And we put these small hairpin RNAs that target either a beta-2 subunit or a beta-4 subunit of the nicotinic acetylcholine receptor into a virus that can infect nerve cells. And then we infuse that into the arcuate nucleus of a mouse. And what you can see here is that when we take these small hairpin RNAs targeted against the beta-2 subunit or the beta-4 subunit, we can reduce the expression of each of those subunits selectively in the arcuate nucleus of the mouse. And when we do that, we can actually go in and we can see that the other subunits are not affected. So these are very selective. So now we have a mouse that's pretty much normal, except either the beta-2 subunit or the beta-4 subunit is decreased in the arcuate nucleus of the hypothalamus. What happens? Well, first of all, we can see that animals that have that beta-2 subunit that was essential for the initial rewarding and activating effects of nicotine still show a decrease in appetite to a nicotine-like drug. So here's the decrease in appetite. It's small, but it's very measurable when you measure food intake in a normal mouse and when you measure food intake in a mouse that has no beta-2 or less beta-2 subunits in the arcuate nucleus. But these mice that lacked the beta-4 subunit had no effect on food intake when you gave them a nicotine-like drug. Well, what does that mean? That means this is a subtype that's independent of the subtype that's important for reward, that's important for a different effect of nicotine on behavior, for this food intake effect. And this subunit, if you recall, is the one that's really important in the autonomic nervous system, the fight or flight system, for activity and arousal. So that's interesting that it's also important for the effects of a nicotine drug on food intake. So why do people smoke despite negative effects on health? As I mentioned, some people smoke to control their appetite. And we know that one way that nicotine decreases appetite is through these alpha-3 beta-4 nicotinic acetylcholine receptors. And through work by subsequent work to the knockdown experiments, we know that they are on POMC neurons in the arcuate nucleus. And that gives us a different subtype and a different brain area for this important behavior driving ongoing nicotine intake in humans. So now I'm going to switch to the final behavior that I'll talk about today. Of course, there are many others. And that is that another reason that, that smokers and vapors report that they use these products that have nicotine in them is to control symptoms of anxiety and depression. And we know from epidemiology that people who are depressed are more likely to smoke, and also people who smoke are more likely to be depressed. So this is not a one direction relationship. There's an intermingling of the people who are depressed and who smoke. And there's a long history of understanding that the upstream molecule that targets these nicotinic acetylcholine receptors, acetylcholine, is important for mood in humans. So back in the 1970s and early 1980s, Janowski and his colleagues gave drugs to humans that increased the activity of acetylcholine in the brain and body by preventing its breakdown. And if you give this drug, physostigmine, it blocks the breakdown of acetylcholine, then individuals who've never had a history of depression actually report symptoms of anxiety and depression. And people who have a history of depression show increased symptoms of depression as well. And so that led to the hypothesis that increasing activity of the nicotinic acetylcholine system may in fact contribute to behaviors related to anxiety and mood. So to start these experiments, we used a similar strategy to what was done in humans. We gave this same blocker, physostigmine, to mice, and we asked what happens to their behavior. And so if we block breakdown of acetylcholine that targets these nicotinic receptors in mice, then we get more immobility in response to a stress. And this is the kind of behavior that has been used to evaluate the effects of antidepressants, because antidepressants will reverse this increase in immobility. And what happens if we then block the increased acetylcholine receptor activity with either a blocker of nicotine receptors or other classes of acetylcholine receptors? Well, sure enough, we block this immobility. And so what you can see here is a mouse that has been given this acetylcholine breakdown blocker. They are more immobile in response to stress. And if we then prevent the acetylcholine action by then blocking the receptors for acetylcholine, and this is a nicotine receptor blocker, this is a muscarinic receptor, another class of acetylcholine receptor blocker, and if you put them both together, in all cases, what you see is these animals are even less immobile than they were before they got these drugs. So blocking the receptors for nicotine can decrease the responses to stress in this evaluation of antidepressant efficacy in mice. And is this something that is completely independent of other antidepressant medications? No, we see exactly the same thing when we use Prozac, fluoxetine in these mice. Here is the increase in immobility in response to stress in normal mice. And if when we give the acetylcholine breakdown blocker, and if we give those mice fluoxetine, Prozac, then we reduce their behavior back to normal levels. So this increase in stress reactivity that we see in mice as a result of increasing signaling through acetylcholine receptors is sensitive to an antidepressant that should have nothing to do with this system. So we do think this is a model that is relevant to humans who may be depressed. So what we did then was to take the same approach that we did in the arcuate nucleus of the hypothalamus, but with a different molecule and a different brain area. In this case, what we did was, instead of giving this pharmacological blocker of acetylcholine breakdown to the whole mouse, we went in and we used a small hairpin RNA to decrease its expression only in the hippocampus, a brain area where we know that acetylcholine signaling is very high and very important for these stress-induced behaviors. And so what you can see is, once again, we could decrease expression of the acetylcholinesterase by using this small hairpin RNA. We could confine it to the hippocampus, this lovely structure that's here outlined with a green fluorescent protein that's packaged into that virus to show us where we've infused this small hairpin RNA. And what we saw was that we absolutely recapitulated the effect of this drug that increased acetylcholine throughout the brain and body by locally increasing acetylcholine signaling just in the hippocampus. So again, we see more immobility in response to stress in these animals with local increase of acetylcholine signaling. We could prevent that by replacing the mouse gene with a human gene that was not sensitive to this small hairpin RNA. So that's a really good control to show that what we're doing is very selective. And this was not only in one model of stress response in the mouse. We could also see it in a social defeat model, which is a stressor that's perhaps a little bit more ethologically relevant. So it's more relevant to a mouse to actually explore a new mouse that comes into its environment. But if that mouse is a bully, a bigger mouse that beats it up, then it will start to avoid not only that mouse, but other mice as well. And so what you can see is that in non-stressed mice, they will go and explore a new mouse introduced into its cage. In socially defeated mice, they will explore that new mouse less. And if we go to a mouse that's just been a little bit defeated, and we look at the small hairpin RNA in the hippocampus, those mice will now, even though a normal mouse would still explore a new mouse, the mice that have this suboptimal defeat plus the acetylcholinesterase knockdown will avoid that new mouse even more. So it's increasing the susceptibility to stress. It's not all of a sudden making the mouse avoid any mouse, but instead, if there's some stress in their environment, these animals are more reactive. And again, the control animals that have the human gene instead of the mouse gene, they don't show any decrease in exploration of another mouse with this subthreshold defeat, this subthreshold stressor. So this all taken together suggests the hippocampus is a really important place for acetylcholine signaling. And does that have anything to do with nicotinic receptors? And so what we've done is to do the opposite strategy. Instead of increasing acetylcholine signaling only in the hippocampus, we've given that drug an increased acetylcholine everywhere, and now used the small hairpin RNA in the hippocampus to decrease activity of one of the nicotine receptor subtypes, the alpha-7 subtype. Again, a different subtype from the other two that I've spoken about earlier in the talk. So what we see is that if we decrease expression of the alpha-7 subtype, we don't see much effect on its own in these stress-induced behaviors. But what we see in normal mice is that we see increased activity or increased immobility in response to stress when we bathe the whole animal in this drug that prevents acetylcholine breakdown. And if we decrease activity of the alpha-7 subtype of nicotinic receptor only in the hippocampus, we prevent that effect of increasing acetylcholine signaling globally. So that suggests that a different type of nicotine receptor, the alpha-7 receptor, is important in the hippocampus for some of the effects of acetylcholine on stress-induced behaviors. And so if this is the case, then we would suspect that too much acetylcholine in the brain might contribute to depression in human subjects. And so we were very lucky in that we helped to validate one of the tracers for these nicotinic receptors in the brain that could be used in human subjects to evaluate how much acetylcholine signaling actually was going on in individuals who were controls or individuals who had a history of depression. And so what would we expect? Well, this tracer binds to the same site on these nicotinic receptors as acetylcholine. And so if some acetylcholine signaling occurred in the brain of a human subject and this tracer was introduced, we would see availability of some of the sites on these receptors for binding, and we'd see a signal in the human brain. However, if an individual who is depressed had too much more acetylcholine signaling in the brain, then more of these sites on the nicotine receptors would be occupied, and therefore there'd be less availability of the binding of this tracer. And that's exactly what our colleagues saw when they looked using this tracer at the brains of non-smokers who were either had controls, no history of depression, or who were actively depressed. And what you can see in these brighter colors is that each of these bright colors represents binding of this tracer to the nicotine receptors in a human brain. You can see that, in fact, there are very high levels in this part of the brain, the thalamus, but there's binding throughout the human brain for these nicotine receptors. As I told you early in the talk, they're very widely expressed. This is the high affinity type. But if you look in the brain of a depressed non-smoker, you have to have a non-smoker because nicotine also competes with this tracer. Very important to remember. You see that there are fewer bright colors, less of this red binding, and that we thought was potentially competition for this nicotine receptor binding in the brains of people who are depressed. We had to verify that. So if we look throughout the brain, there's less availability for this tracer in people who are acutely depressed, suggestive perhaps of more acetylcholine signaling. So how do we validate that? We then looked at postmortem brain tissue where you could wash out all of the acetylcholine. We used exactly the same tracer. And we saw that in control subjects, depressed individuals who died mostly from suicide while actively depressed, and as control, individuals with schizophrenia showed absolutely no difference in the number of nicotine receptors. So it's not that there are more or less nicotine receptors, but once that acetylcholine is washed out, that tracer was the same across these subjects. And then another colleague, Irina Esterlis, did the opposite experiment. In a non-depressed person, if we block breakdown of acetylcholine using this drug, physostigmine, do we compete? And sure enough, in 5 individuals, before and after blocking the breakdown of acetylcholine, you see the same decrease in binding. And that suggests that here artificially increasing acetylcholine signaling mimics what you see in people who are acutely depressed. And it's already been reported that giving this drug that blocks breakdown of acetylcholine increases symptoms of depression and anxiety in at least a subset of human subjects. So taken together, all of this suggests that these mouse models are reflective of an increase in signaling through nicotinic and other acetylcholine receptors in the brains of people who are actively depressed. So this suggests that acetylcholine signaling is not just important in the hippocampus. Here's the hippocampus. We do see more acetylcholine competition in the hippocampus, but there are many other brain areas as well. And so we focused also on looking at nicotinic receptor signaling in another brain area, the amygdala, where we know that blocking nicotinic receptors, and in this case with 2 different blockers, a full blocker and one that just competes with acetylcholine signaling, decreases the activity of the basolateral amygdala much more than it does many other brain areas. And this is important because we know the amygdala is particularly important for responses to stress. And so in mice, we've now taken again the small hairpin RNA strategy, and we've either knocked down that high affinity type, the beta-2 subunit, or this homomeric type, the alpha-7 subunit, locally in the basolateral amygdala. And there we see an decrease in immobility in response to stress across 2 models of stress reactivity and more social interaction after social defeat stress, which suggests that nicotinic receptor activity in this area is important even without the challenge of increasing acetylcholine globally. So we think that nicotinic receptors set the tone for activity of this nucleus that allows animals to have a threshold for reactivity to stressors in their environment. So finally, why do people smoke despite negative effects on health? Well, we know that they smoke to control symptoms of anxiety and depression. And now we know that alpha-7 type nicotinic receptors in the hippocampus and both beta-2 and alpha-7 type nicotinic acetylcholine receptors in the amygdala are important for stress-related behaviors. They're relevant to depression, they're relevant to anxiety. And so that identifies another novel subtype or another subtype of the nicotinic receptors in another brain area in which behaviors that drive smoking are mediated, and showing that it is absolutely as complex as you might expect. There are multiple subtypes in the amygdala, another brain area. There are likely other brain areas as well that are important for the baseline reactivity to stress. And we know now that maybe blocking these receptors would be more effective in combating the effects of stress, and that perhaps the reason that nicotine and tobacco can alter stress-related behaviors is that the short-term activation of these receptors is then followed by a long-term desensitization, turning off of these receptors that leads to regulation of these stress-related behaviors. So I want to close with this model that we have, that there is an optimal level of signaling through these nicotinic acetylcholine receptors that allows us normally to appropriately react to stimuli in the environment that control our behaviors. If we have too little of that signaling, we get dysfunctions of cognition, as in Alzheimer's disease, in which these receptors and this neurotransmitter acetylcholine is lost. If we have too much activity of this system, as we do perhaps in smoking or vaping, or in individuals who have too much acetylcholine signaling, then we might get more reactivity to negative stimuli in the environment, more anxiety and more depression, and perhaps impairments in encoding of memory. And so overall, restoring this balance of signaling through these nicotine receptors is important not only for smokers, but perhaps in general for other individuals. And so I started with the problem: too many nicotinic receptors, too many brain areas, and too many behaviors. And I think you can see that as a field, we've been able to now start to parse out how these many molecules and how the individual parts of the brain map onto these complex behaviors that maintain smoking. And the good news is that a combination of these molecular approaches that I've described and pharmacology in animal models, as well as in humans, has allowed us as a field to make good progress in dissecting these receptor subtypes and the brain sites that are responsible for specific nicotine-dependent behaviors that drive smoking and vaping. And our hope is that medications targeted to highly specific nicotinic receptor subtypes could be useful in helping either to motivate smokers who smoke or vape for reasons other than nicotine reinforcement, such as appetite control or self-medication of anxiety or depression symptoms, to want to quit and to be able to quit. And maybe it will also help treat non-smokers with weight concerns or major depressive disorder in ways that we aren't able to do now. And finally, I want to acknowledge the many people who were part of this work. I didn't name people as they went along, but you can see that there have been many members of my laboratory from the 1990s on to today, starting from my very first postdoc, Vanna Zachariou, and all of these members of my lab to my current lab, who have contributed to these experiments all along the way. And I want to thank our funders who have allowed us to carry out this work, particularly the National Institute on Drug Abuse, as well as a number of private foundations that have, and the National Institute of Mental Health, as well as a number of private foundations. And here's just a photo of my lab from times before the pandemic, when we were still able to get together in person. And I hope that we'll be able to do that again very soon. Thank you very much for your attention.
Thank you, Marina, for that outstanding presentation. And also, I have to especially thank you for doing such a great job of making the link between the molecular nicotine studies and human behavior. I think that is really the strength of the presentation that we just saw. So we welcome questions for Marina. I'm going to start reading through a few questions that have been submitted so far. And Marina, you can provide answers to this. The first question is, are all nicotinic receptors the same, or are some stronger or weaker than others?
That's a terrific question. And I think it's at the heart of why this molecular work is important, because they aren't the same. And not only are not the subunits the same, but the same subunits can combine in different conformations and change the properties of these receptors. So I referred to that very tight binding site for nicotine, which we think is very important because the amounts of nicotine that get into the brain from vaping and smoking are relatively low. So the tighter you bind, the more likely you are to engage that receptor. So that degree of affinity is important, but also how likely it is for the receptor to stay on when nicotine is bound versus turn off or desensitize, very different across the different subtypes. And therefore, you may have initial effects of nicotine that engage one subtype and long-term effects of nicotine that engage other subtypes, which has implications for pharmacology and development of new treatments for smoking and other disorders. And finally, which ions come through that channel, through that pore, through the hole in the membrane differs depending on the subtype. And that's important because there's certain ions like calcium that are especially important in changing the activity of nerve cells over time. So plasticity, making it very different, the smoker's brain very different from the non-smoker's brain, because the neurons in that brain have undergone changes and in their own properties and in connectivity with other neurons. So each of those subtypes contributes to that differently, and therefore intervening in the activity of each of those subtypes may in fact have different outcomes on behavior and on smoking.
Thank you, Marina. Okay, moving on. The next question is, have there been investigations about how other components of tobacco products might impact the interaction between nicotine and nicotinic receptors? For example, do flavorings like menthol increase the potency or activity of nicotine in brain in these various neurological pathways?
Absolutely. And I believe that one of the preclinical sessions in the early part of this meeting addressed some of those issues in preclinical models. So, for example, Brandon Henderson has shown in elegant in vitro experiments that menthol does change the activity of some of these nicotinic acetylcholine receptors and therefore changes their responses to nicotine. Those experiments have been extended to other flavorants and are currently under investigation by a number of FDA centers. Sven Hjort has looked at those questions as well. And the interaction between flavors, but other additives in tobacco and the receptors themselves, I think is really critical. In addition, other components of tobacco smoke that do not necessarily touch the nicotinic receptors themselves can alter neuronal function. So for example, there are MAO, monoamine oxidase inhibitors in tobacco smoke that don't necessarily interact directly with the nicotinic receptors, but if you can change the breakdown of dopamine, then when the nicotine in tobacco binds to these nicotinic receptors, it will have a different effect on the brain. So absolutely, the different additives and components of tobacco smoke can interact with these nicotinic receptors in ways that certainly promote smoking behavior.
Thank you. Okay, the next two questions are about your stress experiments. The first one is, you showed how the hypothalamus is important for nicotine effects and appetite. It's also important for the stress response being part of the HPA axis. Might nicotine effects in the hypothalamus be important in smoking and stress responses as well?
We know that the appetite, stress, and reward systems are all really deeply integrated. And I do think that the hypothalamus may have some role to play in the stress response as well, although we have only looked at that indirectly. One of the things that we were very interested in is whether stress effects on appetite would be synergistic or antagonized by nicotine in an animal model. So as you know, some of us eat when we're stressed. Others actually stop eating when they're stressed. So it's very, they're individual differences. But since nicotine and tobacco can affect both stress and appetite, we asked in animal models, does that interact in the same direction, or in different directions? Turns out, like everything, it's very complex. There were sex differences. So in female mice, we had different interactions than in male mice. And there are time differences. So acutely, there are effects on appetite and stress, but chronically, the effects on stress and appetite interact in different ways. So I think the hypothalamus is likely involved in some of those, but many different brain areas are contributing to that integration.
That's very interesting, the potential sex differences that might exist in that effect too. The next question about the stress experiments is, can you speak to the effects of nicotinic receptors in acute versus chronic stress?
Yeah, I think that's a very important question. And we know that the regimens of nicotine administration that we use in most of the mouse studies are really very acute. That is, we either give them one dose or a few doses, or we use antagonists, which have obviously are blocking their effects. When we look chronically at the effects of nicotine, we usually give it in a way that desensitizes the receptors. That is, we give it in the drinking water for mice so that it's not stressful in and of itself. And we do see some effects of that chronic regimen on behaviors related to stress, but they are not the same as the acute effects. And we think that acute activation of receptors is very different from that long-term adaptation of the system. But I think you're asking something different, acute stress versus chronic stress. And as far as we can tell, the acute and chronic stressors that we use in our mouse models are both susceptible to nicotine, to manipulations of the nicotinic system. So we use a swim stress as an acute stressor, but the chronic social defeat occurs over 2 weeks. Now, that's not chronic in the way that stressors are chronic in humans, but it is chronic for a mouse. And we do see reversal of those effects of chronic stress by nicotinic agents, or exacerbation by changing acetylcholine signaling in the brain. Then I think the next question is whether there are differences in between tobacco cigarettes and e-cigarette aerosols and how they bind the nicotine receptors. So what's really important in tobacco smoking versus the aerosols or gum or other ways of getting nicotine into the body and brain is the pharmacodynamics. So if you get nicotine into the lungs very deeply, as happens with inhaling smoke from a cigarette, then that nicotine diffuses across the alveoli of the lungs. It goes straight up to the brain, and levels in the brain go up very quickly and they go down quite quickly. And that's sort of ideal for a coordinate activation of these nicotine receptors before you have time to desensitize or turn them off. So that route is indeed very addictive because you open all of those receptors in the dopamine system very quickly at the same time. You get a burst of dopamine release, and that's known to be quite rewarding. In contrast, early types of e-cig delivery, so the very early devices that didn't have the same power or the same components in the e-liquid, were not very effective at getting that nicotine down deep in the lungs. There, the nicotine was absorbed across the mouth, the buccal mucosa, or in the throat. And when you absorb nicotine across the skin, what happens is that it goes to the peripheral blood flow, it goes down to the liver, and before it ever gets to the brain, a lot of that nicotine is metabolized by P450 enzymes in the liver to cotinine. By the time that nicotine gets to the brain, levels are lower and they go up more slowly, they go down more slowly. So that's not very rewarding. And it's not surprising, therefore, that as soon as devices were developed that were more and more efficient at getting nicotine into the lungs, early devices were abandoned. And so you don't see people using generation 1 or generation 2 devices because they weren't as rewarding because of how the nicotine gets into the body. So once the nicotine gets into the brain, once it gets into the body, it does bind to these nicotine receptors. But how quickly, how efficiently, and at what level it gets into the brain and how quickly it's cleared makes an enormous difference on whether it's rewarding and reinforcing or not.
Great, thank you. Sorry, I lost you there for a few seconds.
No worries.
But let me ask you the next question, which is, can you talk about the variability of nicotinic receptors in humans and how they differ by demographics like age, sex, and race?
Right. So the biggest difference in nicotinic receptors is across age. So certainly in the aging brain, there are many postmortem human brain studies that show that we start to lose nicotinic receptors as we're older. Very clear with Alzheimer's disease and neurodegenerative diseases that result in dementia, that loss of nicotinic receptors is accelerated. So it's not only that you lose the acetylcholine cell bodies, so the ones that stimulate those receptors, but we know from very early experiments, some I think from Agneta Nordberg's work back in the 80s, that one of the first things that that you can see in a postmortem human brain, if you're looking for it, is loss of nicotinic acetylcholine receptors. So by age, definitely enormous differences, but more so with dementia. But there are some reports of sex differences. They're not large. It's not a consistent difference that a particular brain area has more nicotinic receptors in men or women. Those are more, have more been looked at in animal models where, for example, chronic nicotine, we know increases the number of nicotinic acetylcholine receptors that are found on cells by stabilizing them and promoting their trafficking. And there have been some studies in animals suggesting there's a sex difference to some extent in that stabilization, that change. But those are much more subtle. The bigger differences are by age.
Great, thank you. So talking about the studies you mentioned on dementia, there's a question on nicotine has actions on cognitive function. Can you comment on how these effects might play into addiction?
Absolutely. And I think that they are intimately involved. So a couple ways, right? So what nicotine does in human cognition studies, if you're unimpaired, if you don't have some neurodegenerative disease or other impairment, is that it improves your working memory somewhat. And we're all pretty efficient. So adding something like nicotine is not great at making us more effective, but it can improve working memory a little bit. And that cognitive response may be important while you're doing a complicated task. You may be able to keep more information on board. And so some people do report that they smoke before they do something where they'll have to pay attention or use that information in a complex task. So that is one reason that smokers report that they smoke and that they say that they don't want to quit. They don't want to impair that. However, there's another aspect of cognition. And of course, cognition is, again, a very complex construct. It means a lot of things. But one of the things that nicotine and tobacco does very well, and this is work that was first done by Eric Donnie, Tony Kajula, and Alan Sved and others, is that it is able to enhance the rewarding aspects of things that are only a little rewarding. So for example, if there is a sound that rats or mice like, and they get nicotine at this time that they get that sound, they'll work more for that sound. And that's not cognition so much as reward enhancement. And there have been some lovely studies, I believe mostly by Ken Perkins in human subjects, showing that both smoked nicotine and vaping of nicotine can enhance the reward that people experience from a piece of music that they have said that they like. So that enhancement of something that you already like, making it even better, is also important for forming the association between the act of smoking and the rewarding things that you do every day. So whether it's having a drink with a friend, having a good meal, other rewarding things that you can think of in your life, people often smoke in concert with or just after that rewarding life experience. And so those associations now form so that now when you do that pleasurable activity, whether it is going out with a friend or going into an environment where you smoke, it now triggers the craving for the tobacco product, and that begins a cycle. And it certainly contributes to ongoing smoking. I like to think that that's one reason that environmental tobacco laws are really good, not just to prevent exposure to secondhand smoke for those who don't smoke, but is also potentially very helpful for smoking cessation, because if there are fewer places that you associate with smoking and fewer places that you go into every day, there are fewer places that will induce cue-induced craving and fewer places that will promote relapse to smoking if you do decide to quit. And so those associations, which are partly a cognitive process, an associative process, are certainly potentiated by the nicotine in tobacco.
Thank you so much, Marina. I think we have actually exceeded our time here. That was an excellent talk, and thank you for answering all the questions. And I'm sure everybody is applauding enthusiastically. You just can't hear them, but we thank you for your participation, and congratulations again.